The aim of this procedure is to selectively trace auditory fibers from the peripheral nervous system to the central nervous system in an embryo. This is accomplished by first performing a microdissection to reveal the ventral chondrocranium and identify the avian cochlear duct. The second step is to perform a microinjection into the underlying acoustic ganglion cells and their axons. Next, the sample is incubated in oxygenated artificial CSF for 20 minutes to allow for dye transfer. Finally, a histological preparation and sectioning of the tissue allows for microscopic visualization of the acoustic ganglion central projections.
The main advantages of this technique over existing methods are that it enables selective tracing of eighth nerve auditory fibers with high resolution, allows for rapid dye transfer in live tissue, and permits experimentation at an early developmental stage.
This method can help answer key questions in the field of hearing research. For example, what are the molecules that guide axons to their central targets, or how does the circuitry respond to injury or ototoxic exposure?
First, add artificial cerebrospinal fluid to a 500 ml wide-mouth Nalgene jar until the jar is 2/3 full. Replace the lid, which has a hole drilled into it, onto the jar. Place a stem bubbler attached to a tank of 95% oxygen and 5% carbon dioxide through the hole in the lid and into the ACSF. Turn on the gas and adjust the pressure to obtain a constant stream of bubbles that reach about 1/3 of the liquid but are not forceful enough to cause splashing over the rim.